Hey, good morning, everybody. My name is Baird. I'm an associate professor here at the College of Nursing. Thanks for being here. Inija Miller and I are going to give an update on COVID vaccinations and other therapeutics. Very quickly, we do not have any significant disclosures. I happen to be enrolled as a research subject of vaccinee in the AstraZeneca clinical trial, which is ongoing. Inija Miller does not have any financial things to disclose, and neither do I. We have a few goals and objectives for you this morning. We're going to talk about the basic science, which is behind active and passive immunity. We will look at the two currently approved COVID-19 vaccines in the United States through today. We're going to talk about a couple antibody therapies for COVID-19. And then finally, I'll look at the testing options and pull apart some of the nuances involved with that. And with that, I am going to hand the presentation over to Inija Miller. She is a senior here at the College of Nursing, actually is just finishing up, and she has recently completed the Vaccines and Global Health course here as well. Here you go.
Thank you so much, Dr. Baird. I'm going to share my screen now. So as he stated, my name is Inija Miller, and I am a senior 2 at the college, and I'm going to be discussing the types of immunity. So as most of us know, there are 2 types of immunity, active and passive immunity. Passive immunity is basically when the work is done for us. So an individual is given the antibodies. The antibodies are not produced by our own immune system. This can provide immediate immunity, but patients often have to come in for several doses of the antibodies because it does not last very long. And there are 2 ways that one may receive passive immunity. A newborn baby will acquire immunity through the placenta, or the baby can receive it through the breast milk. And there is an abundance of antibodies in the colostrum, which is the first portion of breast milk that the baby will receive. And secondly, it's received in blood products. So these are your antibodies such as IgG, IgE. We often give these to patients with autoimmune disorders, such as myasthenia gravis. A patient will come in and receive these blood products through IV administration, and they will have, these antibodies will counteract the symptoms that the patients are having. And then we have active immunity. So active immunity is when our immune systems are given time to shine. Exposure to the organism can trigger, will trigger the adaptive immune response. And the immune system will then create antibodies to the germ. And these antigens can be naturally induced. So basically, if one comes in contact with a disease or contracts something, and they are vaccine induced. So vaccine-induced active immunity, we have various types. Live attenuated vaccines is when a weakened form of the microorganism is used to create the vaccine. This is like MMR, rotavirus, smallpox, varicella. So obviously we don't want to give these vaccines to our immunocompromised patients because it can kind of exaggerate the vaccine and give them symptoms of the virus. In inactivated vaccines, a dead or inactivated version of the microorganism is used. Examples of this would be hepatitis, the flu shot, or rabies. Subunit recombinant polysaccharide conjugate vaccines. This is when we use a protein, sugar, or capsid microorganism. So basically there is an outer casing of the germ and it is injected into the body. And examples of this would be Hep B, HPV, shingles, or DTaP. Toxoid vaccines use the disease-producing toxin made by the microorganism, and an example of this would be tetanus. Let's see if I can. And last but surely not least, our newest approach, the mRNA. So it creates immunity by teaching the cells to make a protein or a small amount of the protein that will activate an immune response. So the Pfizer and Moderna vaccines that are being used to to provide immunity to COVID-19, these mRNA are injected into the body and they create a harmless portion of the spike protein that we have all seen all over the internet, found on the surface of SARS. And once this protein is made, the mRNA is then destroyed. The protein will kind of hang out and flaunt itself on the cells, and our immune system will notice this, and it will trigger an immune response and create antibodies to this protein, hopefully providing immunity. And this concludes my bit about types of immunity. I'm going to pass it back to Dr. Baird. Thank you.
Thank you so much, Inija. That was super. And we're really going to need to know that foundational immunity as we go into the treatments and vaccines that we have here for COVID-19. And I will reference that back as we go along. So skipping ahead to that, I actually have a couple different pieces of housekeeping news that I want to mention as we get going into the COVID-19 specific stuff. We are recording this lecture, and I will provide the link to that at the end of this presentation, so you'll have that later on. If you have any trouble with it, please just email me. That's completely fine. My email is on the flyer which announced the webinar, and it will also be on the final slide. And if you have questions, you may notice, because this is not a Zoom meeting, but rather a Zoom webinar, there's a Q&A box. So feel free to enter questions in the preferred way using that Q&A button that's along the row of regular buttons that you have. But if you want to ask via chat, that's also acceptable. We will check that as well at the end of the lecture, and we should have some time for Q&A. Okay, so having covered those different items with basic fundamental immunity, I'm going to talk about the COVID and SARS-CoV-2 specific therapies and vaccines which we have. So we have a pair of active immunity inducers, as Inija was describing. This gets the immune system of our own bodies involved. Those are going to be our vaccines. You may or may not have heard about the passive immunities which we now have available to treat COVID-19, so I'll talk about those. We have a single antiviral medication. I'll give it a little bit of press time. And then finally, the testing, which I mentioned earlier under the goals and objectives. I want to talk a little bit about testing because testing means different things depending on what we're doing. But first off, I know that because these COVID-19, these SARS-CoV-2 vaccines appeared within a year's time, there has been some head scratching, some consternation about just how that feat managed to be accomplished. How did we get effective and safe vaccines out to the public within a year's time? So I wanted to go and look at that and describe what actually happened. How did we accelerate this process? Everyone has heard about Operation Warp Speed. That was the government's plan to accelerate this research and development process. That really came from the CARES Act. The CARES Act was passed early last spring, just after the pandemic struck and shutdowns began, and it threw about $10 billion into vaccine and other therapy research. So that was used to begin Operation Warp Speed, which was this development of vaccines. So having a lot of money on hand certainly helps. About $8 more billion have been used to fund this program since last May, so at least $18 billion total. And what these public dollars did was they enabled a public-private partnership to occur because the US government does not develop vaccines itself. CDC doesn't do that. NIH doesn't really do that. That is done by private companies, which we've seen, Moderna and Pfizer, for example. And it allowed these companies, by having vaccine candidates pretty early, to build infrastructure, to hire people, to ready clinical trials in different phases in a much faster way than they would have otherwise. These companies, with their own dollars, they are going to be business conservative. They're only going to pursue the best candidates that are available. They're going to go in a stepwise and systematic approach. They're going to try to save monies on building out the factories to produce the vaccines only after it seems very likely the products will be approved. Well, this was a very different landscape for them. So that didn't happen in as slow and systematic a way as normal. The companies were able to really count their chickens before they were hatched by having funds given to them when certain milestones were reached. If they had a good vaccine candidate, they could advance much faster. Operation Warp Speed also mandated, due to the public monies involved, that these companies share information with one another. Normally that would not happen very much, but here research was being shared between the companies which were involved in this project. And I'll say too that the SARS-CoV-2 virus did not come out of nowhere. There was a SARS-CoV-1 virus which caused the outbreak in the early 2000s. Some of us remember that. It got into Canada. There is a coronavirus which causes a Middle Eastern respiratory syndrome called MERS, which has been slowly smoldering for years now. And a good amount of basic research had already occurred for those two coronaviruses, SARS-CoV-1 and the MERS virus. So when the RNA for SARS-CoV-2 was uploaded to the internet in January of 2020, researchers already had a fairly deep amount of information on how to develop vaccines for it, because that work was already beginning to happen, had been happening for several years. And then finally, I'll say that we are currently able to do RNA and DNA changes, manipulation in a much more sophisticated and precise way than we were able to do at any time in the past. And that helped us quite a bit as we needed to assemble some vaccines here. So also part of the vaccine development process, I'll say that all of the phases of development took place. The animal phases occurred with first rodents and then primate studies. These do basic immune research to see if antibodies are even being formed. They also do some very basic safety research to see what that is looking like, if the subjects do okay. Then phase 1 trials, which are just a few people, less than 100 who get the vaccine. They're not really looking for efficacy to see if it prevents COVID, but rather to see if the safety profile looks okay, and also measuring blood antibodies to see if those are forming. Phase 2 trials follow with 1,000 patients or more, so much larger. And here they do start to look at efficacy to see if patients are getting COVID or not. And then finally, the phase 3 trials. And these are the ones which got the most attention, which we heard about in the fall. And these are very large trials. Tens of thousands of people are enrolled into them. The Pfizer trial, you can see there, over 40,000 subjects. The Moderna trial, over 30,000 subjects. This is more subjects than were enrolled into the latest Shingrix vaccine trial, which is the current shingles vaccine, which is on the market. So these are not small trials by any means. So once we have the virus and its structure known, then we need a target for those antibodies which we're going to develop with our vaccine. And as Inija described, we have antibodies we want to form, and they need to lock on somewhere to a piece of the actual virus. And this spike protein that you've been hearing about, that has been a very popular target for these antibody and vaccine trials. So you can see this is the spike protein. It's a protein. It's actually 3 proteins which fit neatly together to make this spike. And you can see the side view there, sort of cone-shaped. And you can see the top view, sort of an equilateral triangle. And there is no getting away from this. As Inija mentioned, you see it all over the place. It's even on the Secours website. And here you see that there is, on the right, there's sort of that triangular structure, which is the top part of the spike. And then more towards the edge of that drawing of the coronavirus, you can see it's sort of funnel shaped. So it fits with what we would expect it to fit. Once we have that target, and another reason that spike protein is such an attractive target is because it is on the outside of the virus. It is exposed. The virus needs it. The virus can't get into cells without that spike protein because it enables the virus to stick to our cells and then get into them. So we're not going to see coronavirus without spike proteins. So that's highly unlikely. So it's a good stable target for us to aim at with our vaccine development. So the two vaccines that we have gotten emergency approval for, the ones that we're familiar with, we have the Pfizer-BioNTech product, and then we have the Moderna product. And just to shed some light on that Pfizer-BioNTech, what is that about? Why do we keep hearing about this BioNTech company? Well, they are a small German vaccine development company. And when the code for SARS-CoV-2 first was uploaded over a year ago now, they looked at that and they said, we have a technology, this messenger RNA technology we've been working with, and we think we could make a pretty good vaccine candidate with that. So they got to work on that. Well, once the pandemic began and there was a mad scramble to develop vaccines, Pfizer, which is a very large American pharmaceutical company, $160 billion a year in revenue or more company, they did not have that head start. They did not have much basic research going on right then for coronavirus vaccine development. So they looked around and they found BioNTech and they partnered together. So Pfizer, big company, lots of resources, they did the clinical trial groundwork, they did the paperwork, they knew the regulations of how to get drugs out and onto the market. BioNTech did the basic lab work. Moderna did basically all of their work on their own. But I'll say that another feature of Operation Warp Speed is that really most of the basic science for these vaccine candidates was done by May or June. Most of the time that was spent waiting on the vaccines to come out was really clinical trials. We had to enroll people, we had to wait and see if they got sick or not. We had to wait and look for safety problems. So the majority of time from May until December was clinical trials. The vaccine candidate didn't change during that time. The basic science had already been done in the springtime. So then we have these two candidates which made it into emergency approval. The Pfizer product actually has a generic name, tozanamaran. You can see they kind of sneaked in mRNA there to the end of it. And they actually have a brand name now, Comirnaty. And here in a feat of marketing, they have managed to make a word which they wanted to resonate with community because vaccine, but they also got mRNA in there and they also got co in there for COVID. So they have crammed in multiple meanings into their brand name. Now that is so new that the label is not even on the vials yet, but I expect eventually we will start hearing it referred to as Comirnaty. Obviously right now we're just referring to these products as the Pfizer one and the Moderna one. They're compared one against another on this slide. The dosing regimen is very similar. Pfizer, 3 weeks after the first dose comes the second dose, whereas for Moderna, they went with a 4-week wait. So basically a booster. The second dose boosts the immune response which began from the first dose. I will say that what happens if you need to give that second dose early? Well, CDC has approved up to 4 days early for either one of these. So day 17 or later for the Pfizer BioNTech product versus up to day 24 or later for the Moderna product. And that makes you wonder, well, what happens if you're late with the second dose? Well, nothing happens except you don't have full immunity until the second dose is given. So the full immunity comes a few days, a week or two after that second dose. So basically a delay in vaccination leads to a delay in immunity. So you want to get people vaccinated as early as is possible, but if there's a delay, we don't start over, we don't do anything special, we just vaccinate as soon as possible, and then full immunity is expected a few days after that. So what do we get for this, speaking of efficacy? Well, we've seen the number of 95% for Pfizer, 94.1% efficacy for Moderna. Well, what does that mean? The way they calculate that is you have two groups of patients. You have a placebo group, and we'll talk about Pfizer here, and then you have the active group, or the vaccine candidate group is the second group. You give both of those groups their shots, either the real candidate or a placebo, and then this was a real world trial. The people left, they went about their business for weeks and months, and essentially what had to happen is enough people had to get COVID until they were able to show that there was a statistically significant difference between the two groups, placebo versus active. So in the Pfizer group, there were 196 cases of COVID-19, which were included in their application to the FDA. Well, 185 of those were in the placebo group, 185 out of 196. That's pretty good. The other 11 were in the active group, the vaccine candidate group. And if you do the quick math on that, 95% of the cases were in the vaccine candidate group, and that's where the number comes from. Which may make you think, well, what about those 11 people who got the vaccine candidate but still got COVID? Well, that's a pretty small number, so we can't do a lot with it yet. However, all 11 of those people had mild COVID symptoms. None of those had severe COVID. So for what it is worth, there is probably some protection from the vaccine, even if COVID is, even if someone does come down with COVID after being vaccinated, they probably still have some protection. And this is very similar to what we say with influenza vaccines. You may still get influenza, but still get your flu shot, because if you do get flu, it probably won't be as bad as it otherwise would have been. And from those numbers, 185 to 11, we would expect that many people in the active vaccine candidate group did get exposed to coronavirus, but didn't get sick at all, didn't have any symptoms, weren't infected whatsoever. So that's pretty good. For Moderna, very similar, 94.1%, really not statistically significant. You heard the numbers for Pfizer. Just a couple people, one way or the other, could have moved that from 95% to 94.1%. So no statistical difference. These efficacy claims began 7 days after the second dose for Pfizer, or 14 days after the second dose for Moderna. So that's when they began counting the number of cases of COVID which people were getting. Well, what about the downsides, the adverse effects? Well, they happen. The FDA has 5 grades of adverse effects, 1 being very mild, 5 being death. No one died. There were no 4s in the emergency use application. However, there were some 3s. Now, the 1s and 2s I'll talk about first. These are the common adverse events here on the slide. And you see there's a lot of injection site pain. Lots of fatigue, headache, muscle aches, and so forth. This is what Inija was talking about. As that immune system gets going, we feel it. The body's doing a lot of stuff immunologically, and we feel that. Now, these are mild effects. Most people were not complaining about them. Mild headache, I felt some chills, no big deal. A day or two after they got vaccinated, maybe last for 1 day, maybe for 2. So fairly mild, but they were there. And there is a difference between the Pfizer and Moderna numbers. And I'll say their vaccines are not exactly the same. So there is going to be some difference between that rate. However, the rate also varies based on how you ask people questions. So if you ask someone, did you have any chills? They are more likely to say yes than if you say, did you have some adverse effects after this injection? Because they may not think of chills. So depending on the way questions are phrased and asked, you get some movement in the numbers. And that process was not identical for the 2 products. So they're in the same neighborhood. You see for grade 3, those are fairly significant adverse effects. People were missing work, things like that. Fatigue, headache, the numbers were higher again for Moderna. Again, part of that is probably explained by the way the questions were asked, but the numbers are what they are. Now, I'll say that we need to do our part when we're vaccinating people, and we should not downplay the fact that they are going to feel side effects. They're going to feel adverse effects from these vaccines. It's highly likely they will have injection site pain and so forth. You see there under common adverse effects, and it's possible they will have more significant ones. So we should not say you'll be fine, there are no side effects, because there are. Some places are not vaccinating their entire staff all in one day or within a day or two of one another, because you don't want to vaccinate your entire L&D staff on one day and the next day have half your people not be able to come to work or not be able to lift, things like that. So many places are spreading out vaccinations so that they don't run into that business. Okay, so covered that. I'm going to move on and look at a little more in depth between these two vaccines. There were some deaths in the trials. They all favored the vaccine candidates. However, these numbers are very low, so they don't tell us very much because they were very small. Many of these deaths were from completely unrelated causes, motor vehicle accidents, things like that. So we know that they favor the vaccines. We know there was less COVID in the vaccine groups, but this was really not a mortality-driven comparison. Bell's palsy is something that FDA is looking at. It's interesting. They don't know if it's a real thing or a coincidence, but Bell's palsy, which is a paralysis, usually of the face, usually temporary, did occur some. You see the numbers there. It was more common in the vaccine group. And FDA looked at that. The cases were not clustered around the same sort of time points. That is, they didn't all happen a week after vaccination or they all happened 2 weeks after. They were spaced out over a long period of time. So FDA didn't know if they were related to vaccination or if they were just the regular background rate of Bell's palsy, which happens anyway. So they're watching that. If you have someone who you know gets Bell's palsy or another significant adverse effect, I would encourage you to report that using the FDA's MedWatch system. Easy to Google that, FDA MedWatch, fairly simple to enter an adverse effect. We need to learn more about these vaccines, so I encourage you to do that, particularly with newer products like these. There were some hypersensitivity reactions, not many. The Moderna product had no severe hypersensitivities. They did report some numbers, and you can see there the number 1.5% for Moderna was almost the same as for the active group, because people do get anxious about vaccinations and so forth. The UK reported a couple patients very early with the Pfizer product that had hypersensitivity reactions. These happen to be patients with very strong immune responses. These are kinds of folks who carry around an EpiPen. So UK said, you know, if you have that reactive of an immune system, let's not do this right now, and we'll come back to you. So there were some that were reported. These are very rare, and they were not in the US trials. There were some pediatric patients which were studied, not very many, and they were only 16 or 17 years old for the Pfizer product. They were studied just for safety purposes, not efficacy. So we don't know a lot about these vaccines in pediatrics until the currently ongoing pediatric trial is published. That is happening. There's one for each of these vaccines. So we will learn more later about that. But there have been some younger patients who did get the vaccines. We know, I'm not going to read the stability stuff, but we know that these are, as Inija said, these are messenger RNA vaccines. That means they are a very delicate single strand of RNA. So they need to be frozen until we're just about to give them. Pregnancy has come up in conversations quite a few times. They did not enroll pregnant patients in the trials of the 30 and 40,000 people that I talked about. However, pregnancies occurred in the trials despite not enrolling anyone who was pregnant in the trials. And we know that these pregnancies, most have now been carried to term. Nothing untoward has happened in those cases. You can see the numbers there. We have not studied it carefully. We do not have expectations that there is going to be a problem. I have looked into the rumors that have circulated about pregnancy in the vaccine. I have not seen anything compelling there. So far, so good. And I do know that the OB folks in the United States actually recommend vaccination for pregnant women, although there may not be enough vaccines to go around right yet. And then lastly, how long does this immunity last? That's a big question people want to know. And the true answer is we don't know yet. We can follow antibodies, but we don't know yet what the antibodies are going to tell us. And furthermore, we need to follow how many people actually get COVID. It's nice to follow antibodies, but what we really care about is do people get COVID or not? And it's just too soon. Just like we had to wait for the clinical trials to progress and finish, we just have to wait and see what happens with immunity in COVID cases in folks who've been immunized. There just hasn't been enough time that has passed yet. But so far, so good. We're not seeing a significant number of repeat infections. So I'm moving out of vaccines now. We've got some more vaccines which are on the way. This is current as of today. Now, many of these vaccine candidates will not be approved in the United States. This is a worldwide number, but the approval number is for the United States. But many are being tested in other countries, and there's no plan to test them here. So we will not get 41 more vaccines. We will get some more vaccines, but not that number. The two that are approved, I just finished talking about. You'll see that not every candidate made it to the end of the road. There are candidates which get abandoned. And then I'll point out that different countries, as we have seen, they have their own review boards for vaccines. UK does its own reviews. The EU has a board which does a sort of collective review of new products. So depending on the country, there may be more or fewer vaccines approved. The USA does its own thing through Food and Drug Administration, FDA. So we're at 2. That's where we are right now. I do expect to see more vaccines in the USA in the first quarter of this year, perhaps even more in the first half of this year. There are a couple companies which are pretty close, so we'll see what we get with those. But for the moment, we are at 2. Okay, so moving out of vaccines now, we move into what Inija was describing as passive immunity. And here, in contrast with vaccines, where we give the piece of the mRNA and we build spike proteins and then we get immunity to that. Well, here with passive immunity, we're simply giving the antibodies to people. We have an infusion of antibodies. These are MABs, monoclonal antibodies, and we simply infuse them, and the person who receives them suddenly has immunity, which does not last. It will last for weeks, not months, versus vaccines, which should last months and then years, if not decades. So this is a much shorter immunity which is gained, but if you have somebody who's on the verge of becoming ill, this temporary immunity can be very, very helpful. So these are pre-made immunoglobulins to SARS-CoV-2. This was part of Operation Warp Speed as well, though it didn't get a lot of press. And essentially, we looked at pieces of the virus. Antibodies were made to those pieces. That was then turned into an immunoglobulin, which is a drug form of antibody. Immunoglobulin antibody, as Inija said, same thing. And now we have 2 of them, which are available. The trick with these products is it seems like the sweet spot for giving them is somebody who has a positive COVID diagnosis but isn't that sick yet. So that's tricky because they're an IV infusion. So how do you actually get the IV infusion into somebody who has COVID, but you don't want to put them in the hospital? We should not give these to people who are hospitalized for COVID. It does not look like these are helpful for people who already need oxygen for COVID. But if they're not yet on oxygen, if they're not hospitalized, but they're at high risk for progression, then these seem to be most useful in that situation. But that's tricky because they do have COVID. They do decrease that likelihood of an ED visit or hospitalization. We actually have these approved in children down to age 12, as long as children are 40 kilograms, and then all adults. And as you would expect, we need to give these, if we're going to do it, as soon as possible after the COVID diagnosis. But it's tricky because we need to find a way to get an IV infusion to these people. So let's take a look at them. There are two of them, and their names are a real mouthful. Bamlanivimab is Eli Lilly's product, and then the Regeneron product is actually a combination of two antibodies, casirivimab and imdevimab. So we'll call them the Lilly and the Regeneron products. They're both just single infusions. As you might expect with antibodies, you just give the bag of antibodies and you're done. It has to be IV. We give them through an inline filter because antibodies and other proteins, they can be damaged during transit and so forth. And when they do, they unwind, they unspool, and that leads to infusion reactions. We really want to filter out those chunks of antibodies. That's why we use those little inline filters. So we're seeing places which are doing this in a couple chairs in an infusion center, which are dedicated to COVID patients, or we're seeing some home health companies which are bringing these to people's houses. But it is a little tricky because we need to give this IV infusion, but they have COVID. We're flushing these lines after the antibody bag is done, and we are seeing the usual infusion reactions that we commonly see with all sorts of IV antibodies that we give in infusion centers, like infliximab and those sorts of things. We get infusion reactions. They're usually pretty mild, and the common adverse effects from these products are they're pretty clean, some mild. You see it there, flushings, facial swelling, and so forth, but pretty clean adverse drug profile. So what do we get for all that? What's the efficacy? Well, when they're given to people who are in that sweet spot of having high risk of progression of more severe COVID, but they're not there yet, if these are given early enough before they need oxygen or hospitalization, then the hospitalization rate is about 3%. If they're not given, the control group for these trials, about 10% of those people progressed into severe COVID. So they are effective. They do bring that percentage down quite a bit. And we had about 3,000 people in each of the trials. And it's a fairly robust number, and we have a fairly good idea of what the side effects profile looks like as well. So moving out now away from passive immunity and antibodies, we have one drug for treatment of COVID. It's remdesivir. I won't spend too much time on it. This drug got a lot of press early because it appeared to have some efficacy against SARS-CoV-2, and it does. But this is for severely ill patients, and there are a lot of trials for remdesivir. Brand name is Veklury, and the data is mixed. It's not a slam dunk product. So it depending on the trial that you look at, it may look better or worse than in other trials. But basically, this is a once daily IV infusion. We give a loading dose of 200 milligrams, give 100 milligrams daily for 5 days after that. If patients get worse, then we may go to an additional 5 days, but that is optional. The adverse effects on this are much dirtier as far as data collection than in the other products, because here you have hospitalized sick people. So there's a lot going on. Many are intubated, many are sedated and so forth. So there's a lot happening. You can see the numbers there. It's hard to tease out the adverse effect from the drug versus an adverse effect just due to all the things and illness that these patients are experiencing, but they are there. We did actually get a lot of experience giving this drug together with chloroquine and hydroxychloroquine early in the pandemic when we were using chloroquine, hydroxychloroquine more. We're not really using them now for COVID, but some people are on hydroxychloroquine for rheumatoid arthritis and other autoimmune diseases. We don't give those at the same time as remdesivir. Fortunately, that doesn't come up very often. So now I'm going to move into testing a little bit, because when we say we're testing for COVID, that can mean a few different things. We have a pair of different kinds of tests which diagnose COVID. So we're familiar with those. These are the nasal swab tests where you need to get a swab from either the mouth or the nose, nasopharyngeal area, because you're expecting to find some viral DNA up there, some actual virus in that space. So here we are looking with the first one. The first one is a nucleic acid amplification test, or a NAT test. It's a type of PCR test. This is the one where you get it and they send it away to a lab, and then some amount of time later, days, you get a result back. What we're doing here is we're looking for little bits of viral RNA from the nose or throat. And the lab is able to amplify that and detect it. It's very good at doing that. If there's RNA there, it's very good at finding it. So if you do this kind of test, the PCR test, and you have a positive result, that means it found some RNA. On the other hand, if you have a negative result, then you're probably not infected. That is, the patient was probably not infected. It's pretty good at having a true negative. There are not very many false negatives. We can see some false positives because some people who've recovered from COVID still have some bits of viral RNA scattered around in their nose, for example, hasn't quite washed out yet. So we can find that, or lab contamination can lead to problems with showing us a false positive. That is, the test comes back positive, but there's really not coronavirus present. It's just remnant or it's contaminant. So that's the PCR test. We also do the nasal swab if we're going to do the antigenic test. This is the so-called rapid test. So what we're doing here is a small test has been prepared which detects the antigen of some part of the viral particle. And as Inija was saying, we know antigens are just some kind of piece of the virus. Could be the spike protein, could be part of the shell of the virus, could be any sort of part, doesn't matter. But this test has been coded to find that piece of the virus. And we do the swab, we expose it to the antigen test, and within 30 minutes, maybe an hour, we have an answer. We have a positive or a negative. So that sounds great. Why would we ever do the PCR test if we could do this antigen test? Well, it's got some drawbacks. The big one is you need a lot of virus there. Because we're not amplifying the RNA of the virus like we do with PCR, you need a lot of virus presence. You need active infection. If you're just starting out, or if you're on the tail end of an infection, it can come back with a false negative. It can say there's no virus present, even though there is some. So you need a robust amount of virus for this test to work. If you have a negative test, therefore, if you really want to show someone's negative, you may simultaneously send away the PCR. And if you come back negative on the antigen test, you can say, well, you're probably negative, but maybe you're at the very beginning or very end. If the PCR comes back positive, then you say, aha, yes, that was the case. On the other hand, it's pretty good at detecting. If the positive result comes back, it's pretty good. So if this test comes back positive, patients are usually positive. There are some false positives, but not very many. So it's better at confirming an infection than ruling one out, because you do need a lot of virus to be present. Okay, the last kind of test is the so-called antibody test. Now, this is a blood test that discriminates it between the two diagnostic tests for COVID, and then the antibodies. We're looking to see, was someone exposed to COVID in the past? Were they infected in the past? And now, maybe it will determine whether they were vaccinated or not. You can get positives for antibodies if they were vaccinated. So as Inija described, and we're kind of coming full circle here, there's two basic ways to get antibodies. You can get infected and recover from the disease. That's the hard way. Or you can get a vaccine, in which case your body will make its own antibodies. There are some other ways to get antibodies. If you get those antibody treatments I described a minute ago, the bamlanivimab, et cetera, that gives you antibodies for weeks, maybe maybe a couple months. Or if you get convalescent plasma, that's another way to get antibodies, but those will go away shortly as well. And I'll talk about convalescent plasma in just a moment. But two basic ways to get antibodies. So this test, this antibody test, we test the blood and we look to see, are there antibodies to the coronavirus or not? If they're present, then the test is positive. If they're not present, then the test is negative, and that generally indicates the person has not been infected, has not gotten any of those medications I just mentioned recently, and might mean they haven't got vaccinated. That one's tricky. But based on what I've seen, results can vary based on the antibody test that we're doing, but it's quite possible vaccination will give you a positive antibody test. So be aware of that if you have that done. Now let me backtrack just a little bit while I'm on that thought. If you look at the diagnostic tests of with the nasal swabs, and you think about vaccination, would vaccination give you a positive diagnostic COVID test? The answer is really no. The short answer is no. That should not happen. So these should not be susceptible to that sort of false positive. So back to the blood test for antibodies. Finishing up, I mentioned that antibody therapy or convalescent plasma can generate a false positive or just a positive result. Now that will fade out over weeks and months. It won't last a long term, but it can show a positive result there. And then finally, we don't know how long these antibodies will show up positive. There are a lot of these different kinds of tests. These are the little blood test things. So think sort of a blood glucose test strip situation. But it just depends on which one you're using. Some are more sensitive than others. And so they're not terrible. I can't say in this slideshow whether or not you would have a positive result after a particular length of time or whatnot, because there's so many different antibody tests. But the idea behind the antibody test is to look for prior exposure. Okay, one last item, convalescent plasma. This is essentially passive immunity, where convalescent plasma is taken from someone who has recovered from COVID and has high titers of SARS-CoV-2 antibodies, and then that is infused into someone who's got a positive COVID test result, so they've got a COVID diagnosis. So very similar to bamlanivimab and the other antibody therapies, same idea, usually needs to be given before they're very sick, but if they're at high risk of progressing. So I'm going to stop here so that we can talk about some questions. I will say, check out this slide. There are, if you look at that tiny URL address, you should be able to find the slides in there. I didn't have time to talk about a bunch of different things, dexamethasone that we use, or baricitinib, tocilizumab, some of the other drugs that we're using to treat the immune response to COVID. Didn't have time for any of that. I did put a paper in here on herd immunity if you're interested in seeing what the end of the line might look like for vaccinating large numbers of people. And I actually have a voiceover of that paper that's available at that YouTube link. But just email me if you would like the full paper. So with that, I'm going to look at first the Q&A and see what we have here. So first question. Were all the Pfizer or Moderna vaccinated subjects tested afterwards for asymptomatic presence of the virus, or just those who presented with symptoms? So I will say, Tina, that blood tests were done in these patients to see if they had converted to antibody positive or not. They knew who had gotten the vaccine and who had not. So yes, the short answer is yes. They were doing serial blood tests and they were looking to see who had converted. So I'm going to say the answer is yes to that. We have a question here. Can you say more about the abandoned vaccine? Usually these are vaccines, so I don't know that one specifically, Marcella. Usually there are two reasons why vaccines fall by the wayside. Well, maybe three reasons. First, safety problems. Second, not generating enough antibodies. Or third, the company ran with two candidates and they only really had resources to make one of them work out. And one of the candidates wasn't as good as the other, so they discarded the second one. I'd be happy to get back to you on that though, because I don't know the answer to that. Let's see, next question. Are the vaccines that are being developed currently, are they expected to be an improvement from the two that are already approved, or are they similar? So that's an excellent question. First, I'll talk about efficacy. You can't get much better than 95% efficacy. That's pretty great. So I don't think their efficacy will approach 100% much more because there's just not much room before you hit the ceiling. Side effect wise, maybe they'll be better. We'll just have to wait and see. Overall, the question is, are they also mRNA vaccines? Are they going to look very similar to the two that are already out, or is there a different process? Because you may remember Inija described quite a few different ways to make vaccines. They are generally different processes. Johnson & Johnson and AstraZeneca are both using an adenovirus, which is a completely different way of delivering immunity. So they will look somewhat different for that reason. So there are a variety of different platforms, they call it, or technologies to make the vaccine work. Inija described every different kind. So the new vaccines that are coming out do use a variety of different ways, and they will have a different profile based on that. So improvement in efficacy, can't do much better. Improvement in safety, will there be an improvement in long-term immunity? Maybe. We'll have to wait and see. It's a really good question. Next question here. Will the antibody test be able to differentiate if the antibodies are from SARS-1 versus SARS-2? I was told if the antibodies come back, you should still get the vaccine because antibodies might not fully cover you. So that's kind of two questions. I like it. First question, SARS-1 is not clinically relevant now. There hasn't been a case of SARS-1 in over a decade. So as far as we know, that virus is quiescent. If it comes back, and it didn't spread as easily as SARS-CoV-2, if it comes back, then we'll have to deal with that at that time. But SARS-CoV-1 is not a clinical concern at this time. However, for the second question, if you have a positive antibody test, should you get vaccinated? So that's a great question. And part of that is, what if you've had COVID and you've recovered, should you get vaccinated? The current recommendation is yes. However, you may not want to start for 90 days after you've recovered. You shouldn't get vaccinated if you're actively ill with COVID. And then there's plenty of good reasoning, and the recommendation is do not get vaccinated for 90 days if you've just recovered from COVID, because you should have your own antibodies. Do you need to be vaccinated at all? The recommendation is to get vaccinated. I think that's reasonable. Waiting 90 days lets someone else get that place in line. I think that makes sense while vaccines are scarce, but eventually I think getting vaccinated is a good move. Let's see, next question is if you had COVID, should you get the vaccine? As I said, recommendation is it's okay to wait 90 days. Let's see, next question. If we don't know how long antibodies last or how long they last, why don't we test before antibodies before giving the vaccine, especially since vaccine distribution it's a scarce resource, basically. So that's an excellent thought. And the reasoning is perfectly sound. If you have someone who you know is antibody positive from who knows how long ago, 3 months, 6 months, why not wait? You can do that. The current recommendation is just the 3-month time window. If it was yourself and you wanted to stretch that, there's reasoning there to do that. So I could understand that reasoning. But the antibody tests, I don't know how easy it is to mass to do mass antibody tests right now? That's another question that's within this question is, if you want to delineate the population and say, you people have antibodies already, wait, you people don't, you're first in line for the vaccine, you're talking about millions and millions and millions of people. So how easy is it to scale that? And you have to do blood tests in all those folks and so on and so forth. That's a logistical challenge. The logic is good, but the actual doing it on the ground is challenging. So I can't comment on how far those conversations have gotten, but your logic is good on that, Kate. Let's see, next question. Does it matter how far the swab is inserted into the nares? Some people just do the outer edges, clarify the best appropriate place. So there are multiple swabs for testing for a COVID diagnosis. There are multiple tests, and the different tests they have different recommendations for where that swab needs to go, whether it's just in the mouth, or if it's up the nose, all the way to the back, or just in the front of the nose. I know people have had multiple tests in multiple places. So really you need to follow the directions on the test itself. And there are actually saliva tests, which are becoming more popular because there's plenty of virus in the saliva, as you would expect. That's what we're concerned about with masking. So you could certainly have tests which just do saliva, and those are getting more popular because people are afraid of the whole tickle the brain thing. But follow the directions on the particular test, because different tests do say to do different things. Let's see. What has AstraZeneca done to improve their efficacy? The original report was 70%, 70%. They're basically, AstraZeneca had some paperwork problems. And they also had a fairly significant problem with the amount of antigen that they gave in some of their test vaccines. They only gave half the amount they were planning on giving to over 1,000 people, and then they had to untangle all of that. So the answer is they have not changed the product. They are sticking with the regular dose and trying to ascertain what is the correct amount of antigen that needs to be in the vial to yield a good immunity. And, it may come out lower than the ones that we have already. So in that case, we'll have to figure out how we're going to distribute those, but they won't be precisely the same. It's actually kind of convenient that the two that are out now have very similar efficacy, because no one particularly cares which one they're getting based on efficacy. They're within a percentage point. But if you have one that's all of a sudden 70, people start thinking a little bit differently about that. But they have not changed the product. The USA trial is still ongoing, won't be turned into the FDA probably until February or March. So we're going to have to wait and see what that data looks like. They haven't settled down in revealing that data yet. Let's see, 2 questions. What are you seeing in terms of the proportion of U.S. population that will refuse the vaccine? What impact will that have on herd immunity? So Melissa, I have got an article on the slide in front of you right now, which delves right into that with herd immunity. The amount of herd immunity you need depends on how infectious the organism actually is. Measles, you need a lot of people to get vaccinated because it's so contagious. On the other hand, less contagious diseases, you get herd immunity at a lower percentage of vaccination. And importantly, the efficacy of the vaccine matters quite a bit. If you have a vaccine that's 80% effective, you need to vaccinate more people with that vaccine than you would if you had a vaccine that was 90% effective. That's all broken down in this paper and this voiceover that is right here in front of you. So the impact we don't know what we need for herd immunity, and it depends on which vaccines we have. It's going to be a lot of people. But vaccine hesitancy, where does that come in? I don't know poll data. I'm a terrible person to ask for an anecdote here, but it seemed like the interest got more about when can I get a vaccine versus I'm afraid of the vaccines. The conversation seemed to shift going from December into January, but that may just be me. Second, mutations. Are you seeing anything in terms of escape mutants, et cetera, and needing to repeat vaccines? I have looked at that. So far, so good. I've even looked at what the spike protein looks like with the mutations in it, and it looks very much the same. So far, it does not appear that these mutations are leading to a lack of efficacy for the vaccines, and the experts that's what they're saying. So I'm not too worried about that right now. The efficacy of the vaccine still seems to be pretty solid. Do you think herd immunity will be created with more people vaccinated, although there are many mutations? Kind of touched on that. I don't think the mutations are going to affect us with the mutations we've seen so far. That seems to be the best guess. Deaths with the 2 vaccines, those are in the EUA. Katie, send me a email and I will be happy to pull that out for you. I believe they were described. Do we think we'll need a yearly vaccine? We need more time. Yes, you suggested we may need more time to know that. We do need more time. Can you post the PowerPoint somewhere? It is, the slides are available on this link here on this slide, which is currently shown. So they should be available there for you. I'm happy to share them. And we are at 12:31. So if folks need to go, that is completely understandable. I'm going to stay here and go a little over and try to answer the rest of the questions. But I thank you for attending. And if you'd like to stay on and get the questions, they will be on the recording. I will keep the recording going and then post it for later consumption. Next question, are we looking at adverse effects of people with a previous infection versus no previous infection? I had a significant reaction. People at work who I know had worse reactions. They're real. Let's not downplay the adverse reactions. They do happen. You suggested that, do we know that adverse reactions are more likely if you had the infection? That would be logical. And we did actually see that there are a bit more reactions with the second dose of both of these vaccines than with the first dose. And that's logical too, because these are mostly reactions that the immune system is generating. So when you have essentially the booster from the second dose, a part of the immune system is already laying there in wait with antibodies and T cells. And when it jumps on that new antigen, you get more of a response. So there was about a 10% bump in likelihood of adverse reactions with the second dose as compared to the first. So that would follow that if you had recovered from COVID, you probably had a somewhat higher chance of having adverse reactions from the vaccine. I can't prove that. I'm just saying that would be logical. I don't know if that bears out in reality or not. Next question, should people receive multiple different types of vaccines? If you got Moderna, Pfizer, should you also get AstraZeneca because it is different? We don't know that yet. I think the short answer is right now, not necessary, but we don't really know that yet. I would also say, just from a pragmatic point of view, we should maybe in a year, we'll be thinking about things like that. But right now, we should let everyone get to the front of the line once so that everybody can get vaccinated once before we start doing deliberate repeat vaccinations. That's a good thought, though. It's a good question. And there is a difference. And the flu vaccines are different every year. So there's some logic there as well. Will the current vaccines provide coverage for the new strains? We think so, Nick. We think so. The strains we've seen so far, pretty sure, yes. On down the road, so far so good. Will we need annual injections, annual vaccines? We'll have to wait and see. Simply enough time has not yet passed by for us to know. Can you transmit the disease after getting the vaccine? That's an excellent question. So Do we know, really what we're asking is, can we still harbor coronavirus in our nose and or mouth after having gotten vaccination? And we don't know the answer to that yet either. It's being studied. Early indications are not really. It doesn't seem to have much risk of contagiousness, but we don't have a really solid answer yet. So certainly much less likely of transmission. And you saw all those people who didn't get infected at all after vaccination. So certainly much, much better. Is there any chance? We don't know the answer to that. That's a great question. In the trials, age range, overall health, excluded for health reasons, there was a very good diversity in both prior comorbidities, diabetes, heart disease, age going up to, I don't know what the oldest person in the trials was. They went into the 80s. They did exclude pregnancy. That's an ethics thing because you're exposing an unborn fetus to a investigational product. So we tend to avoid that when we can. That was not a health thing. That was an ethics thing. And was anyone excluded for health reasons? I can't think of any. The only contraindications to vaccination are prior reaction to either the vaccine itself, so like if you have a terrible reaction to the first dose, you probably don't want to get the second one, or a component of the vaccine. There's a polyethylene vehicle in one of them, which some people have trouble with, known to have trouble with. That's pretty rare. So exclusions are pretty few. We don't usually vaccinate people who are acutely ill with COVID, but otherwise we tend to vaccinate unless there's a contraindication. So not many exclusions. We also had pretty good diversity in ethnicity, which was really helpful. That's often overlooked in trials. They did pretty well with these to enroll different ethnic groups. If you contract COVID after getting your first shot, do you need to start over? Or if you're feeling better, can you still get your second shot? This exact scenario came up, and I'm trying to remember what the answer was to it. I can't remember. If you could send me an email, I will track that down, Krista. I don't want to shoot from the hip on that. I'm just not sure. Let's see. Waiting 90 days to be vaccinated if you had COVID recently. Does this recommendation change if illness occurred after the first dose? That's the same question. If you could email me, I'll be happy to get the current recommendation. And I'll reiterate, many of these recommendations are not based on good data. They are recommendations, expert opinion, that sort of thing, because we don't have good data. So CDC is doing their best at guessing, but many of these are guesses. Minority workers disproportionately affected by COVID-19. That is right. Can you speak of efforts made to ensure minorities adequately represented in Pfizer and Moderna vaccine trials? They were represented pretty well, Benji. Was it in line with the population of the USA? It was close if it wasn't there or exceeded. So I was pretty pleased with it. And I would be happy to pull the EUA for you and give those percentages. You might be able to Google that, but if you'd like for me to pull those percentages, I'd be happy to do it. I was fairly impressed with how they did with that. These are just the two trials I described, Pfizer, Moderna. I don't know how the other companies are doing. They haven't released that data yet. Let's see, vaccine administration yesterday, giving it to some vaccinees. How long can people put off receiving the second dose? Are there any risks associated with delaying it? So Jerry, the basic answer is delayed immunization results in delayed immunity. So you can wait an indefinite amount of time. There's no maximum amount of time before the second dose can be administered. And I get it. Some people just miss the appointment. They'll be back next week. There's no maximum amount of time. They can come back anytime they want and get that. The recommendation is still to just give it. We don't start over. We just give the second dose late and that's it. But in the meantime, in the interim, they are not expected to have full immunity. That's the real problem for them. So delayed immunity. Can you post this PowerPoint somewhere? If it's not available in that link that's on the screen, please let me know. Just email me. I'll get it to you. Supply chain distribution. Whew, that's a big one. Any comments on that? My main comments are it's a big lift, and a lot of people are working really hard, and I don't know where we are right now with it. I can't really comment, but I know it is a huge logistical challenge. So let's be as patient as we can and help when we can. Let's see. If you've been immunized, pass the virus to others, I think I did cover that a little bit ago. It's possible. It's much less likely than if you weren't vaccinated that you could pass it on, but it's theoretically possible. We don't know if it really happens much or not. Let's see. I did see, so Kate, you're correct. The Sputnik Russian vaccine and the AstraZeneca vaccine are both adenovirus. I don't know much about collaboration. I think that's mainly scientific collaboration, sharing data so that they can both cross the finish line sooner. I don't know a lot about it, but I do know they are doing that. And email me if you want me to check on that. I'd be happy to do that for you. When will J&J be approved? I think they will submit maybe next month their paperwork. And we saw the paperwork turned around in a week or two for the first couple. So assuming the paperwork looks good, assuming there's not a landmine in there somewhere or a problem, I'm hoping first quarter of this year for Johnson & Johnson. Let's see. Can you get the first dose of one brand and then restart the series with the other brand? Excellent question. That is not recommended, but it is happening by accident or just because people didn't realize it. So if you start a series with one and then the second dose is the other one, current recommendation is stop. Don't get any more. Just wait. Consider yourself to be immunized for now, or the patient to be immunized for now, and maybe there'll be a later recommendation to update that. But the current recommendation is not to start over with the second product or give any sort of third dose. Let's see. When safe to no longer wear masks? Herd immunity, will that be the main factor? So herd immunity, a number of cases are hand in hand. I think that we're going to have to see the number of overall cases drop enormously before we see masking go away. Currently, there's a lot of COVID around. So once we have more vaccinations, more other good things happening to reduce the case rate, then there'll be just less virus around. Fewer cases tend to lead to fewer cases. One begets the other. And at some point, there'll be a tipping point where there just won't be a lot. And then I think more and more people will get comfortable with it. You're kind of asking a sociological question too, because when will the recommendation change versus when will people just stop masking? That's a pretty good question. Okay. I'm looking at the chat log now. All the other questions are done. It looks like most things came in through Q&A. So we covered that. Allison Keeler, if you're still with us, there is the VAERS, V-A-E-R-S. That is a CDC vaccine reporting system. You can go through MedWatch to report vaccine stuff. They'll talk to CDC. CDC does run its own reporting system called VAERS, V-A-E-R-S, I believe it is. And you can directly enter vaccine safety information into that system as well. And email me if that doesn't sound right. I'm pretty sure that's right. Thank you, Jan Ivy. That's very nice. I covered that. Okay, I think we are good. Thank you again, Inija, for partnering up with me. It's great to have someone working on this stuff together. And thank you all for being here this afternoon. I will get this recording tied up and posted. And thanks again for all the interest. I really appreciate it. Yes, the PowerPoint should be in this slides link, which should be there on your screen. Okay, thanks you all.